Welcome, everyone. It's great to have you all here attending this symposium. Today, we will focus on data harmonization in MRI studies. You may ask why. Why should we harmonize data? Well, neuroimaging is now entering an era of big data. Large sample sizes are necessary for a number of reasons, including the study of individual differences, the generation of sufficient power to study complex questions related to genetics, behavior, and psychopathology. Also, large samples help promote robust and replicable science. However, obtaining such large samples in practice often involves data collection at multiple centers with potential differences in various factors, such as scanner models and manufacturers, and possibly acquisition protocols. Therefore, there is a critical need for tools and approaches to harmonize MRI data that can reduce the unwanted variance across sites, subjects, and scanners, while still retaining individual differences of interest. In this symposium, we will provide a broad overview of the nascent but growing literature and approaches to data harmonization in a variety of MRI modalities, including structural, functional, and diffusion MRI. 4 experts in the field will present their recent work today using different approaches to data harmonization. We will also discuss some challenges inherent to harmonizing MRI data. For example, different MRI modalities have very different properties and a very wide variety of downstream summary measures that are used to quantify and compare subjects. These issues make it very challenging to develop universal approaches to data harmonization. Even within each modality, at which level of the data should it be raw or pre-processed that we consider for harmonization is also a difficult question to address. Not to mention longitudinal data with either brain development or disease progression in time. Some of these ongoing issues may not be addressed today in this symposium, but we hope they will pique your interest. And we also hope by the end of the symposium, our audience will have an understanding of the current state of the art regarding MRI data harmonization and learn how different MRI modalities and research questions may need fundamentally different approaches to harmonization. We also hope our audience will be better equipped to employ harmonization to aggregate samples for their particular research questions of interest. Lastly, on behalf of the other 2 organizers, Dr. Michael Harms and Dr. Janine Beisterbusch, we want to thank all the participants and look forward to your contribution to the discussion.
Hello, everyone. I'd like to start by thanking the organizers for this symposium. It's unfortunate that we could not gather together, but I hope the topic today can still catch your interest and stir some helpful discussions. So my name is Haochang Shou. I'm an assistant professor of biostatistics at University of Pennsylvania, and I co-direct the Penn Statistics in Imaging and Visualization Endeavor Center, or PennSIV. Today, I'd like to share a series of group efforts that's developed by PennSIV members over the years for using statistical models to conduct intensity harmonization across multimodal MRI studies. As we are entering an exciting yet challenging era of next generation neuroimaging research, there are many large-scale collaborative consortium efforts such as ATNI, ABCD study, and Enigma study has made it possible for us to access hundreds and thousands of multimodal neuroimaging data from multiple study cohorts. It also promotes the use of meta-analysis that integrate and combine data across available resources and potentially could enhance our ability to discover novel imaging markers. One of the challenges of conducting such analysis is the existence of unwanted technical variability, also known as scanner differences or site differences. It could be due to the scanner manufacturer, the acquisition parameters, the pre-processing pipelines, or simply just because of the systematic measurement error between the scanners. So in the parallel efforts with the efforts of trying to push for a more standardized acquisition protocols and pre-processing pipeline, as well as developing a novel downstream analysis technique, such as deep learning-based tools that are robust to scanner effects. We took an approach that somewhere in the middle, where we take the pre-processed data and trying to use statistical modeling to remove the unwanted scanner variability while preserving biological variation of interest. A couple of years ago, Jean-Philippe Forching in our group, who was a postdoc at the time, identified there's differences between data coming from the two different scanners that are demographically matched. So as evidenced by the MA map showing on the upper left corner here, we can see that the fractional anisotropy measured by voxel from one scanner on average is always higher than the other scanner. Moreover, the level of difference is also dependent upon the actual voxels and the spatial location. So coming from a genomics background, JP was able to adapt a commonly used tool in the genomics literature called COMBAT to successfully remove such differences between the two scanners, as showing by the lower right bottom corner, the COMBAT results here. It's also compared with a couple of other existing imaging intensity harmonization technique as showing the supreme performance with COMBAT. Moreover, if we are looking at the association of the FA map with age, we were able to see that after the COMBAT harmonization, the voxels that are showing to be significant actually has an increased level of the T statistic, meaning higher effect size. While for voxels that are negatively not associated with age, non-voxels, the T statistic number preserved to be around 0. So the power was enhanced while the type 1 error was controlled. Results also showed that there is increased reliability and consistency from findings that was obtained using each of the individual scanner cohorts. So COMBAT stands for combating batch effects, is a regression-based technique. However, unlike the traditional regression type of modeling where a site was just included as a covariate in the model and the correction was done for each of the individual features separately, COMBAT allows the adjustment of the scanner and site differences in both the mean and the variance of the data across features. Also, COMBAT assumes that the parameter estimated for scanner differences in mean and variance across regions of voxels are drawn from a common prior distribution that allows them to borrow information from each other. COMBAT also includes the clinical variable of interest, so the final corrected and adjusted harmonized data can preserve such the biological variability of interest. Since then, COMBAT has been successfully applied onto other modalities of imaging data, including cortical thickness measures, the resting state functional connectivity data, as well as the task-based functional MRI data. In the meantime, our group has been trying to extend such framework and developing harmonization technique towards more complex settings. Here is an example, an effort led by Dr. Christos Stavrakikos from Penn Radiology and myself, as well as trainee Raymond and Pomponia, where we were able to harmonize a large-scale lifespan data set that consists of over 10,000 baseline structural brain MRI data. The efforts was under the iStaging consortium led by Dr. Davasico, where we are hoping to pull data from multiple resources across lifespan and trying to understand the aging process from adolescent to older people over a wide age range and in a multi-dimensional fashion. The data we are trying to harmonize is the region of interest volumetric data that was obtained through a hierarchical brain partition. And one of the challenges that we are facing with respect to such a harmonization is the unbalancedness of the sample size and age coverage across the 18 studies that was included in lifespan data sets. Also, it covers a wide age range from 3 to 96 years old, and we've evidently have seen a large variability in terms of variance and mean across one study to another. So Ray has developed a nonlinear version of the harmonization technique called the combat GAM, which is based on the generalized additive model. It allows a simultaneous correction of the scale and location differences, but in the meantime also estimates an age trend for each of the region of interest over the lifespan age range. So over here, I'm showing a couple of examples of the age trajectory that we obtained using the combat GAM harmonized data, and the trend are generally consistent with the previously reported literature. For example, total gray matter volume are showing a much rapid decline over adolescent phase and a gradual decline later on, versus the hippocampus volume seems to have an accelerated atrophy in later half of the lifespan. Moreover, we are showing that the harmonized data using combat GAM can also help with prediction, the brain age prediction features by reducing the mean absolute error in each of the cohorts. Another product from this project is a web-based Shiny app that Ray has developed, which allows the interactive visualization of the estimated age change for a user-selected region of interest. The user can also upload their own data set towards our server and have the data to be harmonized with the lifespan large-scale data as a reference. In the meantime, Joanne Beer, who is a postdoc trainee in Pensive and worked with my colleague Kristen Ling and Takeshi Nohara, has also developed a longitudinal version of the harmonization technique that can handle within-subject correlation of the repeated scanner and also enhance the estimation of the time effects. Lastly, I just want to mention one of the recent developments in terms of looking at harmonization by not just looking at the scanner and site differences in the mean and the variance of individual feature, but also looking at potential scanner differences in their covariance structure. As we are moving from mass univariate analysis where each of the individual features are treated as independent, there's an increasing need of looking into multivariate pattern analysis where the covariance structure between features can play a crucial role. So the scanner differences, if they exist, can have an impact in the downstream results. Andrew Chen, who is a PhD candidate in our group, and together with myself and Takeshi Nohara, we look at the cortical thickness measure obtained from the ATNI-1 study that was obtained using the ANT cross-sectional pipeline. So Andrew indeed finds the scanner differences in the covariance matrices between the Siemens and GE scanners. He then proposed a novel harmonization technique called the COVSPAT, which not only corrects for the mean and the variance difference, but also improve the correction of the scanner differences that exist or present in the covariance structures. When we were experimenting the before and after harmonized data and in terms of how they're doing in differentiating between sites, as well as predicting clinical meaningful variables such as the disease diagnosis and gender, we did see that the co-vet methods were not able to differentiate between different scanners, while the unharmonized data can do the scanner detection almost perfectly. And co-vet also further improved on the performance in detecting patients with Alzheimer's disease versus controls. So co-vet essentially is a two-step algorithm that was built upon the COMBAT. And the detailed methods can be found in the virtual poster number 1249 that will be presented by Andrew. Lastly, I want to mention a couple very exciting directions that our group are moving along the same line, including one of the freshly funded grant proposal submitted by Pensive, where we are hoping to extend and further develop novel harmonization methods that can adapt it to more complex study design, including longitudinal nested design and hierarchical design, and also improve the corresponding performance in machine learning and NDPA in a more distributed fashion, so the data don't need to be shared across center in case security is an issue. There's also a parallel effort led by Dr. Ragini Verma at Penn, looking into harmonized connectomics data sets from the DTI measure, helping us to understand the autism disease among kids. The software and the algorithm are available on GitHub and in languages including R, Python, and MATLAB. Lastly, I want to thank you all for listening and thank all my collaborators and Pensive members. As I said, this is a completely group effort developed over a couple of years. And this is a picture of our Pensive last year over our retreat at New Hope, Pennsylvania, when social distancing wasn't a thing yet. Hopefully we can go back to our normal routine very soon. Thank you, and I'm happy to take any questions.
Hi, my name is Hatton, and today I'll be talking about multi-site data harmonization, particularly during a longitudinal study of childhood and adolescence, the ABCD study. I will briefly be talking about the study and then the standard bias that we can see in the data, and then some of the processes that we're looking at for normalizing T1 and T2 data, as well as a dynamic DTI atlas that we're developing. And finally, I'll close out with some of the harmonization considerations that I would like to raise with the audience. I'd like to acknowledge that there's thousands of people involved in the collection of the ABCD data, and I'm just fortunate enough to be able to share all their hard work with you today. The ABCD study assesses around 12,000 children, ages 9 and 10 for 10 years, and it's across 21 sites in America. And every 3 to 6 months, they have a questionnaire or cognitive assessments. And biannually, every 2 years, they have an MRI and biospecimens acquired, such as DNA. The data is publicly available at the NIH. We started off by harmonizing the MRI acquisition by making sure that it was based on the PING study and the Human Connectome project, so that we could generalize between the three projects, which would be able to generalize more broadly. We collaborated with the scanning manufacturers to ensure that we had high temporal and spatial resolution across the manufacturer's 3T systems. However, and also more in-depth discussion on how we pulled all the data from the 21 sites into a central data processing center to ensure that the processing was done in the exact same manner is highlighted in a very lengthy paper by Don Hagler that was released last year in Neuroimage, and there's also a copy on bioarchives of a draft for those interested. Despite all these protocol harmonization approaches, we still do see some between scanner differences. On the left here, we've got the T1 grey-white contrast is lower in the Siemens, and we can see that GE is slightly different in mean cortical thickness and cortical surface area than the other manufacturers. when we're looking at DTI, we can see that Phillips is offset in FA in mean diffusivity and neurite density. And this is typically because Phillips has a tendency to null out the background noise to zero to increase signal to noise ratio. And this needs to be considered when comparing diffusion sequences between manufacturers. So I'll start off with the T1, T2 intra-individual intensity normalization. I'll be talking about a T1, but this is applies to T2 as well. So what we wanted to do is looking at the ping T1 protocol, we wanted to vary the flip angle and the acquisition style and the inversion time and see what happens with the intensities of the voxels within the brain of human phantoms. And with test-retest, you can see on the left that the profiles of the intensities of the voxels and per count are exactly the same, which is great. But then when we change the flip angle down, there's a swing to the lower intensities, and flip angle up, we move the intensities more to the right. And I'll take your attention to the last figure on the right, inversion time. When we reduce that, as in we darken the voxel intensity, we kind of dim it down, you can see that intensity, there's a lot more lower, darker intensity voxels, and that the peak is much reduced to the right there. So what we've been doing is, after a basic N3 correction, is transforming the intensity profile so that they match the target profile, which is the ping protocol all the way on the left. Why is this important? Because when we look at the low TI inversion time, we can see that FreeSurfer has quite a difficulty in being able to distinguish the grey-white peel surfaces as opposed to on the right, which is the optimal parameters used in the ABCD study and the ping study. So this also translates to what FreeSurfer thinks is that the lower the inversion time, that the brain is smaller, and we can see with N3 correction that there's still a large displacement between the optimal scan and the lower inversion time. We added white matter bias correction because in these adolescent youths there's quite a high intensity occipital signal that we needed to suppress for better segmentation. But what's important is this intensity normalizations of the voxels. We're able to really salvage a lot. And I must say that this is mainly cortical. Subcortical structures are much better at intensity differences between scanners and parameters. So interestingly, we weren't really able to change much of the goodness of the fit, but the bias, the volumetric differences between the target and variations in protocol were able to be resolved just by this simple inter-individual intensity correction. Going on to DTI harmonization, we started looking at the 1,300 subjects from the ping study, and we got the averages of the FA RGB maps to be able to show that on average, this is a 3-year-old female's brain, and then we go to an 8-year-old brain, and onto a 20-year-old brain, and there are changes, very subtle but important changes over that period. So if on the right, the corpus callosum in particular changes in size and shape over this period. So what we are working on is a multispectral data atlas, DTI atlas, which follows the eigenvectors and eigenvalues rather than a voxel intensity. And this is a multidimensional DTI atlas because we also have age-specific atlases within it and sex-specific atlases within it, and then an offset. We're working on an offset for manufacturers' platforms. So that's something that's in the pipeline. we've just finished the acquisition of the second time point for MRI, and so we're working on an even larger data set to be able to be generalizable and shareable. Other harmonization considerations I wanted to raise with the audience today was between sites, and this is a lot of work done by Enigma, is that clinical scales can be different between sites, such as a depression scale with HAM-D and Bex. How are they equivalent? That's an important consideration when you're looking at multiple sites. Similarly, drug dosage, such as clozapine equivalency, if you're looking at psychotic disorders. The Enigma group has done a great amount of work on these harmonization problems, and so I direct them to their website to tell you about these sort of ways of being able to balance different scales and different dosages. Longitudinally, this is an age while we're looking at young childhood through to adulthood, where there is a large brain trajectory change, and we always need to keep into consideration, are the brains going in the trajectory that we expect or not? So that's being able to QC your own results to ensure that they're biologically sane. Practice effects with task fMRI. We did a study with a aging VETSA, the Vietnam era twin study of aging, which showed that men that came back after 5 years could still remember the task, and it's very important to address practice effects in longitudinal studies. These kids are doing these tasks every 2 years, and they're still having cognitive improvements during that period. So how to understand practice effects and normal adolescent cognitive development with respect to task fMRI is important to be considered about. Our questionnaires are age appropriate. So to ask a 9-year-old about psychotic-like experiences is different to if you're asking them at 16 or 20, and similar with sex-related questions or drug usage. So they need to be harmonized so that they are age-appropriate questions for the subject, and yet they are equivalent across that time span. Participant identification. Sometimes there's mislabeling of subjects. If there's 2 subjects in one day, they might have the ID mixed up. And it's important to ensure that the follow-up brain scan is the same brain as the previous scan, taking into consideration that the brain is growing in this period. So we use mutual information registration to be able to get a score to check any outliers to ensure that they are the same person. And of course, it's very tricky when we have twins and triplets being scanned as well. But we think we're doing a really good job. Participants can relocate to another site, and I don't know the answer to how you harmonize with that. Just something to consider in the design of your study. Similarly, we have had a lot of dental appliances, particularly braces, which provide, which are safe for the MRI, but do not give usable data. So we had a drop of about 5 to 7% just because of braces in the follow-up scan. So you need to consider that when looking at the power when setting up your study, that there will be dropouts for dental appliances. Scanners will be upgraded by software and by hardware. You need to test those changes to ensure that they don't affect the core parameters. And with that, I'd like to thank you for listening, and I'd like to acknowledge all the wonderful partners, as well as the ABC families and child participants. Thank you.
Today, I'm going to talk about our strategies on diffusion MRI data harmonization, which is critically important for the joint analysis of multi-site studies. I have nothing to disclose. Large-scale data sets are important to boost the statistical power in neuroimaging studies. However, we cannot naively pool diffusion MRI data across multiple scanners for joint analysis. The slightest differences in hardware, software, or sequence of the scanners can induce much larger unwanted and nonlinear scanner effects on the diffusion signal. For example, when the same subject scanned across two scanners with a similar sequence, we observe significant differences, around 10% difference in fractional anisotropy in white matter, about 20% in gray matter, and up to 35% difference in CSF areas. This example acknowledges the common agreement that scanner differences can cause substantial unwanted effects on the diffusion signal. The efforts to remove the unwanted scanner-induced effects from the diffusion MRI data are termed harmonization. A harmonization method can be considered successful if the harmonization preserves the biological variability of each subject while removing only scanner-related differences. There have been multiple attempts for harmonization of multi-site diffusion MRI data. We can categorize the methods into two. First type of methods are data pooling techniques, which aim to statistically remove the scanner differences from the derived diffusion measures. Harmonization is performed in the last step of the pipeline of the statistical data pooling techniques, and each diffusion measure is separately harmonized. Second type of approaches remove the scanner-specific differences from the raw diffusion MRI signal, which includes also multiple work of ours. The approaches in this category are applied at the signal level early in the preprocessing pipeline and accounts for nonlinearities in the scanner bias in this diffusion MRI signal itself. Therefore, the method allows the use of any diffusion MRI data processing like tractography and model fitting after harmonization. Depending on the availability of the traveling subjects, two primary approaches have been proposed by our group. First one is retrospective harmonization. It is for use where data has been already acquired without traveling subjects. Therefore, it uses age and sex match healthy controls to learn the scanner differences. Second one is prospective harmonization, which includes more ideal and state-of-the-art harmonization techniques, but it requires traveling subjects scanned at each site to create a within-subject overlap cohort. Although the approaches in retrospective harmonization can be also used here, more advanced nonlinear strategies such as deep learning should be preferred for more accurate harmonization. To capture scanner differences, we propose rotation invariant spherical harmonics features using spherical harmonics coefficients. Rich features represent the energy of the signal at different frequencies. They also represent the different microstructural tissue properties which are independent of the orientation. In the figure, you can see rich features of the same subject scanned with two scanners. In our strategy, we select one scanner as reference, and the other scanner to be harmonized or mapped to the reference is termed as target. The retrospective harmonization includes two parts. First part is learning part. The learning part is performed using training data. That's a subset of healthy controls that were age and sex match across two scanners. The aim of the learning part is to create the scale maps of the voxel-wise average rich features of each scanner, which are later used in the harmonization step to the target data to appropriately modify the diffusion MRI signal. On the other hand, for prospective diffusion MRI data harmonization, we develop deep learning-based techniques to learn the nonlinear mapping across two scanners using traveling subjects. I will explain the details of prospective diffusion MRI data harmonization in another talk. If you're interested, then please join me on Tuesday. We have applied the retrospective diffusion MRI data harmonization method in multiple applications. One of these was a large schizophrenia database collected from 13 studies. By leveraging our harmonization method, we were able to characterize white matter pathologies across the schizophrenia lifespan. In doing so, we identified 3 distinct clusters of fiber tracts, and we were able to show the dynamic interaction of the pathologies at different ages and stages of the disease. Given the importance of the multi-site harmonization problem, 2 harmonization challenges were organized in the past years. These challenges provided a fair platform to evaluate the performance of the state-of-the-art harmonization techniques. 2 tasks were evaluated in the most recent challenge to compare the performance of the participated 21 algorithms. Our approach was selected as the best performing one in multiple categories with the minimum error from the ground truth. In the graphs, you can see the performance of all algorithms for fractional anisotropy and mean kurtosis measures of white matter. Lower error represents the better performance, and our results are highlighted by yellow. As part of an already funded NIH R01 grant, our team is working on a very exciting project on harmonizing multi-site diffusion MRI data from 30,000 subjects belonging to the ABCD study, Healthy Brain Network, and Human Connectome project. Harmonized diffusion MRI data will be shared with the community using the NIMH data archive. We will also develop a formal ontology-based system for defining 189 white matter fiber tracks using neuroanatomical landmarks known from human and monkey literature on brain connectivity. Finally, we will develop a web-based system for real-time 3D viewing and querying of the harmonized data and fiber tracks for a user-defined selection of subjects from the entire cohort of subjects across different diagnostic categories. And this system also will be integrated with NIMH data archive infrastructure. Before concluding my talk, I would like to emphasize the important points and challenges that need to be considered by all harmonization algorithms. First of all, any harmonization method must ensure that they do not introduce any new information in the data while harmonizing. To demonstrate this, in our earlier work, we ran a tractography experiment to compare the anatomically segmented white matter tracks before and after harmonization, and to observe the harmonization effects on the fiber tracks. So we observed significant overlap between the tracks before and after harmonization, which means that we haven't introduced any new fiber orientations during harmonization. Second, validation of the harmonization should be done on multiple metrics, including several model-independent metrics, as well as a few widely used ones, such as fractional anisotropy, fiber orientations, mean diffusivity. If the data is multi-shell, mean kurtosis and RTOP can be also calculated. We recommend being cautious against multiple points while using statistical data pooling techniques for harmonization. First one is potential preprocessing effects or software bias on multi-site diffusion measures. The slightest changes in the preprocessing pipeline can add bias on diffusion measures, which will eventually add bias to the harmonization of the statistical data pooling techniques. To demonstrate this, we preprocess all diffusion MRI data using the same preprocessing pipeline, but only computation of FA differed. We observed significant software effects in estimation of FA in multiple sites, as seen in the figures. The slightest changes in the preprocessing pipeline added bias on the diffusion measures. Therefore, we highly recommend using the same preprocessing pipeline across the sites in multi-center neuroimaging studies. The other challenges for statistical techniques include pooling datasets with very different p-values, very different resolutions, as well as understanding whether statistical covariates can truly model nonlinear scanner effects. To summarize, we demonstrated the significance of inter-scanner reproducibility problem for multi-site diffusion MRI data analysis. We also presented our retrospective harmonization method, which is model-free and allows the use of any diffusion MRI data processing after harmonization. Using rotation invariant spherical harmonics features in harmonization, we ensure that we do not introduce any new information in the data while harmonizing. Finally, our retrospective harmonization software is open source and available online. It supports both single shell and multi-shell harmonization. If you're interested in, please check it out. This is the end of my talk. I would like to thank to my mentors, my team, and my collaborators, as well as our fundings which supported this work. Thanks for listening. I will be happy to answer the questions.
Hello, everyone. My name is Blake Dewey. I will be presenting today on the harmonization of structural MR images from using deep learning strategies. Let's start today with a case study. So we have here an MS patient who went through annual scans from 2006 actually to the present, but we have here 3 scans from that time period. And we can see here that as technology improved over that time, we were able to increase our SNR while maintaining the same contrast. But we also have here disease progression that is co-founding the actual technical improvements here. So we actually have 2 things working at once that might be not so beneficial for our automated processing techniques, as they have to distinguish biological and technological related differences. We have here even a larger change as a scanner upgrade and protocol change was enacted in 2017. So we have a completely different T1 weighted MP range contrast than we did previously. These consistency issues are especially evident on 2D sequences such as this T2 weighted flare. We have here in 2008 a low resolution 2D flare. We have in 2011 a flare that was corrupted by motion artifacts. And then 2016, a completely new 3D flare that has a different contrast as well as increased resolution. You can see how this could cause a little havoc in our automated processing as they try and distinguish biological actual changes from these scanner-related changes. So our solution here is going to be deep learning, specifically supervised learning, using training data to predict our outcomes here. Each network is going to contain millions of parameters, and in a specific way, we're going to use the UNet strategy, the UNet architecture. The UNet was originally designed for biological segmentation, and it uses downsampling to collect coarse and fine features at the same time. We're going to modify that a little bit for harmonization from the original UNet by Ronenberger et al. That you can see here on the right. Now, first, before we actually start training our data, our network, we have to get training data. We acquired imaging of an overlap cohort of 12 subjects, 10 with MS and 2 healthy controls. We scanned them on 2 different scanners with less than 30 days between the scans. You can see an example subject, actually a healthy control here, on both of the scanners, scanner number 1 and scanner number 2 for each of the 4 contrasts that we're going to be looking at: flare, T1 weighted images, PD weighted images, and T2 weighted images. This is our actual trained network. The architecture that we have here is very similar to the UNet that I showed you before, with one specific difference. We have a skip connection here between the input images and the final feature maps that's actually going to change this into a more residual network, where the network only has to predict the changes in the images. And this is beneficial here because we expect the outputs to be very similar to the inputs in a lot of different ways, because they are only being harmonized. We're not actually changing anything about the structure of the image, so we shouldn't have to recreate them. We train this on 128 by 128 voxel patches, and we use multiple contrasts as channels, and then predict all of our images at once. You can see this goes from 4 to 4. So we use 4 contrasts as inputs, and we output 4 contrasts that are harmonized. In pre-processing, we use SMORE, which is a self-super resolution technique, and that allows us to improve the through-plane resolution of 2D acquired images. We also co-register our images and then apply white matter peak normalization so that we are aligning the actual absolute values of the white matter between images and image types. We can see here that there is a small difference between the image quality of the pre-processed only images from scanner 2 and the harmonized images from scanner 1. Now, these are meant to be identical, but harmonized images are different from acquired images. They benefit from multiple acquisitions, they tend to have less noise, but they also tend to be a little bit blurrier. So we're actually going to want to create a harmonized version of scanner number 2. And I'll show you how we do that. Now, we've already said here that all images must be harmonized, even if the contrast does need to change, and this is, of course, in the case of scanner 2. So on the left here, we have our original network. We take scanner number 1, run it through network 1, and produce an approximate harmonized image. What we want to actually do is also train a separate network where the inputs are from scanner number 2, and the target is the synthetic images created through network 1. And this allows us to produce a harmonized image from scanner number 2 that doesn't actually change the contrast. Let's look at some qualitative results we have here at T1 MP-RAGE and T2 FLAIR from both pre-processed and harmonized versions of Scanner 1 and Scanner 2. Specifically, we can look at the lesion depictions in the pre-processed images, where we can see that there's a slightly different appearance of the lesions in this particular subject between scanner number 1 and scanner number 2. But in the harmonized version, they're very similar. And in the T1 weighted MP-RAGE, we can look at the cortical ribbon, which appears to be much smaller in scanner number 1 than in scanner number 2 on the pre-processed only images. But remember, these were taken very close to each other, so there should be no biological difference here. Now, when we look at the harmonized images, we can see that those are nearly identical in their qualitative appearance. Then also we can look at the cortical segmentation of these T1 weighted MP-RAGE images. We can see, first of all, that there are some artifacts created from missegmentation of the cortex in scanner number 1, but it doesn't appear on scanner number 2 in the pre-processed version. But in the harmonized version, this is severely or is significantly decreased. We can also see that the cortical ribbon is overestimated in the scanner number 2 image than compared to the scanner number 1. In the harmonized version, however, there's a much closer overlap. To put this into numbers, we calculated volume bias. So this is a percentage difference in volume between scanner number 2 and scanner number 1 for our overlap cohort. And in scanner number 2, we actually show positive values here when scanner number 2 is higher. So we can see a significant, indicated by bold values, bias in each of the measured areas: cortical gray matter, cerebral white matter, thalamus, lateral ventricle, and intracranial volume. But in our harmonized versions, not only have we reduced the bias substantially, but it is also no longer significant when compared to 0. When we look over time in a longitudinal cohort of our MS patients, we can see here that in our pre-processed version, the blue, which indicates scanner 1, follows a fairly normal downward trajectory that we'd expect from atrophy. But once we transition to scanner 2, which is in orange, we see a substantial increase in all cortical gray matter volumes compared to their last scan. We know that this can't be true for all these subjects. But when we look at our harmonized version, we can see that not only is the grouping and consistency of the downward trajectory much tighter, but now the orange values also follow the same downward trajectory for almost all patients. Future work for this topic will include unpaired strategies to eliminate the overlap cohort. We have an accepted paper at Mackay 2020 that will tackle this topic. We want to improve the realness of synthetic images, and we want to do a larger evaluation on downstream tasks, specifically segmentation when looking at public data sets and large multi-site studies that we are a part of. So in conclusion, we have a small overlap cohort. Remember, only 8 subjects were used for training, and this allows us to harmonize structural MR images. So we show that there's a substantial improvement over pre-processing alone when looking at segmentation results and direct image correlation. And this will allow longitudinal studies to continue even after a significant change to protocol or hardware. And I can show you here another example of these T2 weighted FLAIR images, where we go from 2008 to 2017. We have low resolution flares, flares with motion artifacts, and even a sequence change involved. But we can see that after harmonization, these images are much more similar. We now have a very consistent image background to be able to segment these accurately and consistently regardless of scanner. I'd like to give a special thanks to all of the collaborators and funding that we received for this project, and as well as give my contact information, my email here, for any questions that might pop up.